Question 11, 2 to go. In 1991, the body of a man was found frozen beneath a glacier in Italy. Jeez, poor bloke. The researchers named him Otzi. What a name. It was determined that Otzi died 5,300 years ago and that his body is the oldest mummified human body ever found. Scientists have successfully extracted DNA, so we know we're looking at something to do with DNA, from the nucleus of his frozen cells. So what we want to do is describe the process scientists would use on a small sample of Otzi's DNA to obtain larger quantities of identical DNA. Now, what do we do when we actually want to go from a small sample of DNA and we want to amplify it to larger quantities? We use the classic polymerase chain reaction. And that's my first dot point. You don't really need to include it, but you're using polymerase chain reaction, or PCR. Then essentially we want to go through these steps. Now, I will preface this by saying that this is a lot of just memorizing and just knowing these steps and applying it to Otzi's case. Obviously, if you want to learn how to memorize information more efficiently and fruitfully, go back to the week of videos that was actually in the 5-week biology challenge. But I will state here that the best way really to remember something like this would be simply to articulate it many times, but especially to someone else so they understand. You could actually construct a dialogue with them and just say, what is the first step of PCR, second step, third step, and so on. But in this way, if you articulate that particular sequence of steps many times, maybe 3 or 4 times, then you will absolutely know this and it will be ingrained in your memory and you won't have to really go over it any more than that. Without further ado, let me delve into the steps of it. The first thing that we know we want to do with PCR is to heat Otzi's DNA to approximately 90 degrees. Obviously, there's 90, 92, 93. I've just gone for 90 here. But you want to cool the DNA mixture. Why do we cool the DNA mixture? We want the primers to attach, and we cool it down to around 50 to 55 degrees Celsius. Then the third step is we reheat it to 72 degrees Celsius, and we add Taq polymerase, which we know extends those primers, and we add those nucleotides to expand the primers. And the whole purpose of this step we know is to copy the strands, and then we repeat this process many times because we know that it is exponential. If I get rid of that highlighter and we go back here, it's 2 to the power of n, where n are the number of times that you actually perform this, and we know that becomes exponential. So 20, 30 times on, you've got a lot of copies of that exact DNA, and that looks at the process of PCR. Remember, just nice clear steps, show them your logic. There's no need to use full sentences here, just nice clear dot points. Okay, now we move into the next part, and now what we know is that they're using gel electrophoresis. We know gel electrophoresis is used to separate DNA according to its molecular weight, or you will call it size, and obviously it's charged. We look at the scientists discovered that there were 4 different types of blood on Otzi's clothes. He's been up to a bit of mischief. So we look at this, and this is the particular sample of gel electrophoresis that we've gone through. If I look through this, which blood sample on Otzi's clothes belongs to Otzi? Obviously, we're just looking for what matches up with the actual sample. So we know this are the actual samples. What matches up identically? Sample 1 doesn't, sample 2 doesn't, sample 3 is the winner because it's exactly the same. Therefore, we look at number 3. So the next one is to propose one hypothesis, so you don't have to go on with many, that would explain the presence of other blood samples. So the other blood samples on Otzi's clothes, I've just gone for the clothes might have had other people's blood on it. I know you could have answered with animal blood, but just some way in which there could have been an explanation for all the different types of blood that were present on it. So this next question was actually answered quite poorly. We look at it and only 13% of students got 2 out of 2 marks, which shows that there was not really a thorough understanding of the way in which amino acids or the amino acid sequences relate to evolutionary relationships. So when we look at this, it says the analysis of mammalian proteins provides evidence of evolutionary relationships. Explain how amino acid differences in a protein can indicate evolutionary relationships. So what are we looking for is amino acids. If amino acid sequences differ a lot, we know the species are not really related, or they're less likely to be related. So when we look at this, if amino acid sequences are actually similar, then we know that they are more related. So what we know is that over time, as it progresses, mutations occur. So my first statement would look at the idea that mutations accumulate that may change the sequence of amino acids over long periods, obviously to do with a lot of ideas of natural selection, etc. But these mutations will occur. if these mutations occur, we know that the sequence of amino acids are becoming different. So, the greater the number of differences present in those amino acid sequences, the less related the species are, because we know it comes down to the genome. And we look at these ideas of amino acid sequences. Large differences in amino acid sequences means they are less related. If they look like they've got, I suppose, a lower amount of differences, so they're more similar, therefore the more related, or the more closely related the species are. And so we've just got to really work on the idea that 13% only got this correct, with obviously we look at 50% actually getting 0 marks as well. So this is a really key question that looked to differentiate between students. Okay, last part D. So the development of bipedalism in hominins, obviously being on 2 feet, has believed to have a very significant effect on human evolution. Okay, describe 2 effects, so only 2, that effects of bipedalism on hominin behavior. So keyword behavior. We're not looking at the physiology of anything or the anatomy. We're just looking at how it's actually affected behavior. What could they actually do as a result of bipedalism? And the 2 I've gone for is, we know if they were on all 4s, then they couldn't actually make these tools. So my first one was it freed the hands for tool making. You could have obviously looked at the idea of carrying the young as well. And the second one I looked for was it enabled hominins, bit of a typo, hominins to raise their head to a high point to scan for predators. You could have actually looked at the idea of cultural behaviours such as ceremonies, actually to perform these particular things, or reaching higher food as well, only looking for 2. But the key thing you really wanted to look for here was behaviour. What did it actually enable them to do? A lot of students, it says in the assessor's report or the examiner's report, looked at physiological ramifications.